Dear students, welcome to this program. Today we are discussing an important topic entitled Subcellular Fractionation, Differential and Density Gradient Centrifugation. Before discussing in detail the subcellular fractionation, let us first know its objectives. The objectives are to understand Introduction to Subcellular Fractionation, Centrifugation and its principle, Differential Centrifugation, Density gradient centrifugation and its types. Now let us begin with Introduction to Subcellular Fractionation. Living organisms have been grouped into two main classes, Prokaryotes and Eukaryotes. Prokaryotes are primitive organisms, have relatively simple internal cellular structure. In contrast, eukaryotic cells have a complex internal structural organization. Their protoplasm contains a variety of well-defined membrane-bound compartments like nucleus, mitochondria, lysosomes, Golgi bodies, endoplasmic reticulum, centrioles, vacuoles, and ribosomes that can be identified under an electron microscope. Such membrane-bound bodies are known as subcellular organelles. Functional studies of these organelles and other macromolecular complexes require detailed observation and manipulation, which is impossible with whole cell. One of the most fruitful approaches towards achieving this goal is subcellular fractionation that permits the isolation of specific cellular organelle and allows us to work out biological processes free from other interfering reactions in the cell without having to worry about keeping the cells alive. Now coming to the cell fractionation technique. The procedure of cell fractionation comprises mainly of three distinct steps. Step first, homogenization of tissues and cells. Step second, isolation of different subcellular organelles using centrifugation. Step third, analysis of isolated fractions. Now let us discuss the important aspects of these three stages of cell fractionation separately. Step first, homogenization. Before differential centrifugation can be carried out to separate different portions of a cell from one another, the cells and tissues samples must be first homogenized. For the optimum homogenization of tissue specimens, mincing of tissue has to be performed in presence of a biological buffer system that exhibits the right pH value, salt concentration, stabilizing cofactors, and chelating agents. The optimum ratio between the wet weight of tissue and buffer volume as well as the temperature and presence of a protease inhibitor cocktail is also essential to minimize proteolytic degradation. There are different methods available for breaking open the cells depending upon the type of experimental material. Some of them are grinding, cutting with blenders, sonication, and osmotic shock. Grinding with abrasives. Grinding in a mortar and pestle in presence of sand or alumina is a useful method for disrupting bacterial or plant cells. Cell walls are ripped off by the abrasive. However, this method is appropriate for handling relatively small samples. Cutting. High-speed mixers such as varying blenders are also used. Disruption of cells occurs due to immense shearing force generated by the fast rotating blades. Tissue is cut into small pieces and blended in presence of buffer. This method is ideal for disrupting mammalian or plant tissues. Sonication. This method is ideal for a suspension of cultured cells. It involves breaking open the cells using high-frequency sound waves. Sound waves cause disruption of cells by shear force and cavitation. Osmotic shock. In this method, cells are broken by subjecting them to osmotic shock. Cells are allowed to swell up and explode by placing them in hypoosmotic buffer. Now let us discuss the step second of cell fractionation, that is isolation of subcellular organelles. Crude tissue homogenates containing organelles, membrane vesicles, and other structural fragments are divided into different fractions by centrifugation in a supporting media which is usually close to isotonic. Sample is placed in tubes that are inserted into a ring of cylindrical holes in a metal rotor. Rapid rotation of the rotor generates enormous centrifugal forces which cause particles in the sample to sediment. The rotor chamber is sealed and evacuated. This reduces friction, prevents heating of the rotor, and allows the refrigeration system to maintain the sample at 4 degrees centigrade. During centrifugation, the particles differing in their shape, size, or density sediment at different rates under the influence of applied centrifugal force and hence get separated from each other. Since subcellular fractionation, the topic of our main focus, is absolutely dependent on centrifugation, so we need to first understand the theoretical aspects of centrifugation. First of all, the principle of centrifugation. When a particle in suspension is rotated at a high speed, it is subjected to a centrifugal force G, given by the equation G is equal to omega square r, where r is the radial distance of the particle from the axis of rotation and omega is the angular velocity of the rotor. The angular velocity of the rotor in radians per second is given by omega is equal to 2 pi into s dash divided by 60, where s dash is the rotor speed in revolutions per minute, and hence the centrifugal field can be expressed as G is equal to 4 pi square into s dash square into r divided by 3600. The centrifugal field is more commonly expressed as relative centrifugal force RCF, which is the ratio of the centrifugal acceleration at a specified radius and the speed to the standard acceleration due to gravity, and can be calculated from this equation. RCF is equal to capital G, that is centrifugal acceleration, divided by small g, that is acceleration due to gravity, is equal to 4 pi square into s dash square into r divided by 3600 into 981. On simplifying this equation, we get RCF is equal to 1.119 into 10 raised power minus 5 into s dash square into r. Under the influence of applied centrifugal force, particles migrate at a rate that depends not only upon the applied centrifugal field but also on the nature of the particle, that is, its density and radius, and also the viscosity of the surrounding medium. The rate of sedimentation, sometimes called sedimentation velocity, is given by the equation V is equal to dr by dt is equal to m into 1 minus nu bar into rho divided by NAF into omega square r, where r is radius at which the sedimenting particle is located, t is time, m is molecular weight of the particle, nu bar is partial specific volume of the molecule, that is inverse of the density of molecule. Rho is density of the solvent, F is frictional coefficient, omega is angular velocity, Na is Avogadro's number. This equation simply states that the rate of sedimentation of a given particle is proportional to the molecular weight, the applied centrifugal force, and the density difference between the particle and the solvent, and inversely proportional to the frictional coefficient. That means the more dense a biological structure is, the faster it sediments in a centrifugal field. The more massive a biological particle is, the faster it moves in a centrifugal field. The denser the biological buffer system is, the slower the particle will move in a centrifugal field. The greater the frictional coefficient is, the slower a particle will move. The greater the centrifugal force is, the faster the particle sediments. The sedimentation rate or velocity of a biological particle can also be expressed as its sedimentation coefficient, given by the Swedberg's equation. S is equal to dr by dt into 1 divided by omega square r is equal to m into 1 minus nu bar into rho divided by NAF. This equation indicates that sedimentation coefficient is dependent upon the molecular weight, the density, and the frictional coefficient. The sedimentation coefficients of biological macromolecules are relatively small and are usually expressed in terms of Swedberg units. 1 Svedberg unit equals 10 raised power minus 13 second. Two different approaches employed for separation of subcellular organelles from tissue homogenates are differential centrifugation and density gradient centrifugation. Differential centrifugation. Differential centrifugation is based upon the differences in the sedimentation rate of biological particles of different size and density. Large dense particles sediment faster than small light particles. Material to be separated is divided into different fractions by successive centrifugations at increasing applied centrifugal forces. Initially, all particles of a homogenate are evenly distributed throughout the centrifuge tube. During centrifugation, they move down the tube at their respective sedimentation rate. The large particles form a pellet on the bottom of the centrifuge tube, leaving smaller sized structures within the supernatant. However, during the initial centrifugation step, smaller particles that are present near the base of the tube also get pelleted, causing a certain degree of contamination. To minimize the cross-contamination, pellets are usually washed several times by resuspension in buffer and recentrifugation under the same conditions. Resulting supernatant fractions are recentrifuged at a higher speed and for a longer time to separate medium-sized and small-sized particles. Differential centrifugation techniques are conveniently employed to isolate intact organelles, for example, mitochondria from liver cells. The repeated centrifugation at progressively higher speeds and longer centrifugation periods divides the liver homogenate into distinct fractions. Typical values for centrifugation steps are 10 minutes at 1000 G to pellet nuclei and cellular debris, 20 minutes at 20,000 G to pellet a fraction enriched in mitochondria, and 1 hour at 1 lakh G to separate the microsomal and cytosolic fractions. The fractions prepared by differential centrifugation are not pure. Each fraction is enriched with particular component, but it inevitably contains significant proportions of other organelles as well. For example, during fractionation of liver cells, the mitochondrial fraction is often contaminated with lysosomes and peroxisomes. Density gradient centrifugation. Density gradient centrifugation is used to separate macromolecules that differ only slightly in size or density. During centrifugation, the solution tube is stabilized in the tube by a density gradient. The gradients may either be of discontinuous or continuous type. Discontinuous or step gradients can be prepared by either overlayering or underlayering technique. In the overlayering method, known volumes of solutions of decreasing density are carefully allowed to run slowly down the side of tube to form layers over each other, beginning with the densest step by use of a pipette. Alternatively, in the underlying method, the gradient can be formed beginning with the least dense step by depositing each layer at the bottom of the centrifuge tube through a pipette placed with its tip at the bottom of the centrifuge tube. The more dense solution displaces upwards the layer of less dense solution already present in the centrifuge tube. Continuous density gradient is the more common of the two and requires use of a special instrument known as a density gradient mixer. In continuous gradients, density changes linearly with increasing distance from axes of rotation. Two different methods employed for separation of subcellular components on density gradients are the rate zonal technique and the isopycnic technique. In rate zonal centrifugation, the sample under study is layered on top of a preformed liquid density gradient, the highest density of which does not exceed that of the densest particles to be centrifuged. Under centrifugal force, the particles will begin sedimenting through the gradient in separate zones according to their relative velocities. That is, macromolecules with the same sedimentation velocity move as a single zone. Separation of similar type of particles is based mainly on their size. Since the technique is time dependent, the centrifuge run is terminated before any particles reach the bottom of the gradient. After particle separation has been achieved, the density gradient and the entangled particle zones are usually removed from the tube and collected as a series of fractions. This may be achieved in several ways. One of the oldest and simplest methods is to puncture the bottom of each centrifuge tube and allow the contents to slowly drip out. If the bands can be visually detected, the gradient can be recovered simply by carefully lowering a pasture pipette into the band and slowly withdrawing the required material. A method of choice for most applications is upward displacement. A denser solution of the gradient medium is either pumped through a long needle passing through the gradient to the bottom of the tube or introduced via a hollow needle piercing bottom of the tube. The gradient is displaced upwards and the fractions are removed in sequence using a pipette or syringe. Now the isopycnic centrifugation. The isopycnic technique separates particles into separate zones according to their buoyant densities and not according to their shape or size as in rate zonal centrifugation and is independent of time. The size of the particle affects only the rate at which it reaches its isopycnic position in the gradient. Separation in this technique involves the combination of sedimentation and flotation. The sample is layered on the top of density gradient column, the maximum density of which must exceed the density of the most dense species to be separated. During centrifugation, the sedimentation of particles occurs until the buoyant density of the particle is same as the gradient material. Once equilibrium has reached, sedimentation stops, irrespective of how long the centrifugation continues, as the particles are floating on the cushion of material of higher density. It's important to note that the runtime cannot be shortened by increasing the rotor speed. This only results in changing the position of the zones in the tube since the gradient material will redistribute further down the tube under greater centrifugal force. Alternatively, the sample to be separated is initially mixed with the gradient medium to form a solution of uniform density. Under the influence of the centrifugal force, the gradient redistributes to form a continuously increasing density gradient from the top to the bottom. This variant of isopycnic centrifugation is referred to as isodensity method. The macromolecules of the biological sample, which were initially uniformly distributed, seek an area in the tube where the density is equal to their respective densities. The macromolecules now either sediment or rise to this region of equal density. Stable zones or bands of the individual components are formed in the gradient. The purification of mitochondria-enriched fraction obtained after differential centrifugation can be effectively accomplished using this method, since these subcellular organelles differ in their densities. After density gradient centrifugation, mitochondria are recovered as a major band. Lysosomes and peroxisomes get separated from mitochondria and also from each other as two distinct bands. Finally, the third step of cell fractionation, that is, analysis of fractions. Fractions are analyzed to identify and quantify the purified fractions so that they can be used successfully in downstream applications. Different methods employed for analysis are light or electron microscopy of the separated fractions, determine presence of marker enzymes, assay for a protein marker with an antibody, that is immunoblotting. Determine the protein concentration by using a spectrophotometer, for example, Bradford assay. Determine specific activity, that is the ratio of activity of the enzyme of interest to the protein concentration. Testing for marker enzyme represents the more traditional way of characterizing subcellular fractions following centrifugation. Other analytical methods are useful for the characterization of subcellular fractions when simple and fast assay systems for testing marker enzyme activities are lacking. Routinely used enzymes as subcellular markers are the sodium-potassium ATPase for plasma lemma, glucose 6-phosphatase for the endoplasmic reticulum, galactosyl transferase for the Golgi apparatus, succinate dehydrogenase for mitochondria, acid phosphatase for lysosomes, catalase for peroxisomes, and lactate dehydrogenase for the cytosol. With this, we conclude our today's lecture. Hope you have enjoyed and understood it very well. Thank you.